Magnetic resonance guided gene therapy is beginning to revolutionize how to treat rare genetic brain diseases. MRI is enabling a shift from merely slowing disease progression to actually curing these diseases. Infants with ADC deficiency are unable to produce dopamine, leaving their brains trapped in an infantile state. Intraparenchymal infusion targeting the substantial nigra and ventral tegmental area has recently been shown to correct this genetic mutation. The Food and Drug Administration has approved Kebilidi by PTC Therapeutics TM to promote dopamine generation, enabling normal brain development. The treatment is transforming catatonic infants into normal toddlers, as reported in this case by Cincinnati Children's Hospital. In most genetic brain diseases, the genetic error eventually causes damage to the entire brain. In lysosomal storage diseases, for example, gene therapy will require vast brain coverage to create a cure. Here we see a guided treatment in the lysosomal storage disease known as Batten disease, where considerable damage has already occurred in this 10-year-old. Successful treatment will likely need gene therapies at much younger ages. This paper describes how current and expanding development in MR diffusion imaging will likely us to characterize infant brains in ways that enable earlier treatment. Adenine-associated viral vectors, AAVs, and nanoparticles serve as the genetic trucks used to deliver genetic correction. While ultrasound mediation can allow permeability of the blood-brain barrier in local areas, it is immunologically and economically infeasible to deliver a gene therapy large enough to correct large regions of the brain using systemic delivery. Instead, pressurized delivery of AAVs or nanoparticles is achieved through minimally invasive catheters. This pressurized delivery is known as Convection Enhanced Delivery or CED, as it creates effective bulk, convective movement instead of ineffective diffusive movement. AAV vectors, approximately 20 nanometers in size for modest genetic payloads, can effectively convect through the brain's interstitial spaces, which are about 50-100 nanometers wide. Flow rates are quite slow, on the order of 5-20 microliters per min, to allow the buffer carrying the vectors to move through the interstitial spaces without damaging the brain or backflowing up the catheter exterior to CSF exit pathways. Multiple catheters in each hemisphere delivering infusate over a day or two will likely be needed to cover large volumes of the brain. This paper describes why local infusions started in gray matter start expanding spherically, but when they reach white matter, they tend to advect preferentially along the white matter tracts. The large-scale organized orientation of white matter makes it a much more attractive region for convective flow than gray matter. Thus, a neurosurgeon can't just look at catheter locations and estimate where the final infusion volume will distribute. Forward modeling of likely end infusion distributions for various initial catheter positions is thus essential to know which parts of the brain will be treated by each catheter. BrainLab navigation system have provided a predictive distribution product called iPlanFlow to estimate likely distribution volumes. These algorithms are based on the theory of flow through a poroelastic media, here the brain. Non-invasive mapping the hydraulic parameters needed for these algorithms is impossible. However, slides 8-11 detail how they can be estimated from clinical brain MRI clinical catheter placement. Today is largely done in the magnetic resonance MR suite using ClearPoint SmartFrame TM. Analysis of Convection Enhanced Delivery clinical trials revealed two types of trials. In trials that aim to treat a local gray matter region, the delivered clinical dose is often quite close to an estimated interstitial volumes. For Parkinson's disease, MR guidance has refined surgical techniques to reliably fill the desired treatment region, often the putamen. In aromatic L-amino acid decarboxylase deficiency, the substantia nigra and ventral tegmental area trials demonstrate higher treatment percentage. Malignant glioma trials use larger doses to ensure complete coverage of targeted brain volumes. In trials for diseases needing much larger coverage, analysis often reveal large mismatch between infusion volumes and brain regions requiring genetic correction. Fearing that the white matter tracks will preferentially steal the treatment design, pharmaceutical companies have often opted to treat only specific white matter tracks and accept incremental clinical outcome improvements. For instance, in NCL Batten disease trials, the clinical dose for the white matter tracks is far from fully covering the estimated interstitial volume and are largely leaving gray matter untreated. As stated earlier, neurosurgeons can't estimate the end infused distribution simply from visualizing the initial spatial locations of the catheter. Instead, this slide shows how clinical MRI is used to create surrogates that map the hydraulic parameters needed to predict the expansion of the infusion. This slide shows how MRI allows us to characterize the desired and deleterious pathways for infusion expansion. MRI allows us to segment gray matter, white matter, and CSF. The next slides demonstrate on why characterizing white matter is so important. Hydraulic conductivity is crucial for predicting infused distribution derived from magnetic resonance imaging surrogates. In gray matter, the pore fraction is approximately 20% before Convection Enhanced Delivery infusion, with minimal variation during Convection Enhanced Delivery due to its disorganized nature. In contrast, white matter with its organized myelinated tracks can realistically double its pore fraction during convection-enhanced delivery, enhancing infusate distribution. Here we show a video on how infusate races along the white matter tracks. Utilizing diffusion tensor imaging, DTI, one can estimate hydraulic conductivity tensor from DTI maps. As shown earlier, estimates of the pore fraction under pressure scale the translation of DTI to hydraulic parameters. This parameterization is key to using Darcy's law to building the forward model to predict CED distributions. On the left side, we see how enhancing Sulci representation shows where the low-pressure sinks exist to stop CD advancement. Here we use technology often used in MR cardiac sequences, balanced steady-state free precession to create bright fluid maps. On the right, the Sandy-Matlichel diffusion tensor imaging model is used to estimate the extracellular volume fraction distribution across brain regions. Normally a forgotten parameter in Sandy modeling, the extracellular volume gives a direct estimate of the water compartment in which the infusate can travel, the water outside the axons or extra-axonal water. This is a prime example of how advanced diffusion MRI methods can directly be applied to advanced gene therapy guidance technology for whole brain diseases. Once the infusion pumps are running, real-time magnetic resonance monitoring of the growing infusate distribution is critical for refining the final infusate distribution prediction. This allows for flow adjustments during procedures, increasing the chances the target regions will be covered. Transitioning to MRI-guided convection enhanced delivery (CD) in infants involves leveraging general myelin characterization and high-resolution anatomical imaging. This approach aids in adapting gene therapy distribution predictive model with characterization of white matter development in infants before brain disease cause brain damage. The MR community is urged to explore advancements that enhance understanding of CD infusions in infants' brains.